This is the Green Planet Monitor. Voices and stories from a warm, wet planet. I'm David Kattenberg. Today, drug-resistant infections, the new pandemic, and a tangled network of tiny tubes pulsating beneath our feet. Fungal networks below ground sustain life above ground. It's a staggering statistic. This past May, U.S. officials announced that COVID has killed 1 million Americans. The number is likely higher. Globally, over 6 million have died since the pandemic began in January 2020. Meanwhile, a potentially deadlier enemy is sweeping the planet. Bacterial pathogens that are drug-resistant. According to a recent report in the medical journal The Lancet, in 2019, drug-resistant bugs were linked to 5 million deaths worldwide. 1.25 million deaths were directly attributable to them. According to a UK government study, by 2050, antimicrobial resistance, or AMR, could kill 10 million annually. As with COVID, drug-resistant bacteria are not equal opportunity killers. Marginalized folks are the hardest hit. I spoke with two experts in the field. Shira Daron is an associate professor of medicine at Tufts University in Boston and the director of the Antimicrobial Stewardship Program at Tufts Medical Center. In 2021, she co-authored a letter to the journal Nature Medicine entitled Antibiotic Resistance, a Call to Action to Prevent the Next Epidemic of Inequality. Shira Daron joins us from Boston. Tomislav Mestrovic is an associate professor at University North in Croatia and a scholar at the Institute of Health Metrics and Evaluation at the University of Washington. He's worked closely with the authors of that Lancet study on the global burden of drug-resistant infection. Tomislav joins us from Seattle, Washington. I'd like to ask you to start off, is the drug resistance crisis receiving the media attention it deserves? And is it a crisis? Is it a global crisis? Shira Daron, why don't you start?
Yeah, it is a crisis, and it isn't receiving the attention it deserves. You know, people like me who work in the hospital and take care of patients with complicated infections, we see it every single day. And increasingly, we see patients who have infections for which we have no antibiotics. We have nothing in the arsenal. And yet it isn't in the media. And why is that? Maybe because it isn't novel. Maybe because it's been happening very gradually and worsening over time, and therefore it doesn't get the same kind of attention as something like COVID-19, a novel virus, obviously, that can affect everybody. And maybe it's because people don't feel like it touches them, but it will ultimately touch people in all walks of life eventually. And that's why these calls to action are so important.
And it's amazing, like 160,000 deaths in the United States each year attributed to or directly linked to antibiotic-resistant infections. 160,000 deaths each year.
Yeah, and we feel that. And that, to me, that's not a surprising number, given what I see every day. You know, I wonder if people think there will always be another antibiotic developed by pharmaceutical companies, but I don't think we can count on that. I know we can't.
Tomislav Mestrovic, your thoughts on this?
Yeah, I completely agree with what Shira has said, and AMR is definitely one of the biggest threats of this century, and also poses the threat not only to human health, but also to animal health, to food, to our environmental security, and of course global economy also. But the news media coverage and how the AMR is conveyed or framed in the media can then powerfully shape public perception of AMR risk. But as Shira said, in many countries, news coverage on this issue is very sparse, and articles commonly represent AMR as a social problem that it's mainly caused by, for example, dirty hospitals or by some others who misuse or overuse antibiotics. But the role of many important social actors is neglected. The role of social determinants of health that have a role in AMR are marginalized. And a lot of media coverage revolves around new scientific discoveries related to AMR and AMR solutions. But as Shira mentioned, this is scarce. New antibiotic won't be always available. And these sometimes put individuals in the position of ignorance, and then they lack agency for the AMR agenda. And maybe sometimes how the stories are covered may even undermine the public's motivation for taking collective action to tackle AMR, and maybe sometimes even can trigger like feelings of helplessness, paralyzing individual actions. So this is something that we have to take into account, especially now when we know the full scope of this problem and of this issue. We have to work on the public understanding of AMR and exactly what drives it forward.
Shira, Doron, and Tomislav, what drives antimicrobial resistance? So we're talking about bacteria that are out there. They're common, common species, common strains, but the emergence of strains that are resistant to some or numerous or even all antibiotics. We call these superbugs. What drives the proliferation of antibiotic resistance? What makes it happen?
Well, mainly it's antibiotic use, antibacterial drug use. And I like to say it isn't overuse of antibiotics that cause resistance, it's use of antibiotics that causes resistance. And some of it we have to do, some of it is absolutely necessary. So we better not do the antibiotic use that isn't necessary. And so that antibiotic use is happening in healthcare settings, actual antibiotic prescriptions. It's happening when people take unprescribed antibiotics. It's happening when people give antibiotics to animals, both agricultural and companion animals. And then it's happening in the environment, runoff from farms where antibiotics have been used, or even just to a small extent, people dumping unused antibiotics into landfills, et cetera. And then there's a little bit of use in plant agriculture as well. So there are just so many places now where we're essentially pumping antibiotics into our environment, changing human and animal microflora under that evolutionary pressure. And that's only going to get worse over time.
Because when you expose bacteria to antibiotics, you know the ones that don't die proliferate. What doesn't, kills them, make them stronger. Some of them, they just naturally develop mutations that make them resistant. Where use of antibiotics is selecting for resistant strains, isn't it?
That's evolution 101. The bacteria are going to try to survive, and they will mutate accordingly.
And so let's take this apart a little bit. Like you're talking about misuse of antibiotics. Break this down a little bit for us, like Tomislav, like using antibiotics to treat viral infections, or broad spectrum versus narrow spectrum, and all this. Just basics.
Yeah, really important points that Shira has covered.
Run through this.
Yeah, and when we talk about misuse or overuse sometimes of antibiotics in humans, that pertains to, for example, not finishing a course of antibiotics, and like you mentioned, taking antibiotics for viral rather for bacterial infections. And then also,
because viruses are impervious to antibiotics.
Yeah, it's completely different. So bacteria are prokaryotic cells that we can use some of their structures to target with our antibiotics that viruses just don't have. Viruses have to enter human cells to reproduce. So this is a completely different organisms. And this is why antibiotics won't work for viral infections. But also, Shira covered some other important points of general use of antibiotics. For example, we sometimes even in whole states and countries have mass drug administrations, which means regular provision of antibiotics to a large group of people to treat an infection, regardless of whether individuals are ill or not. This is one also of important drivers of resistance.
Go through this for me. What happens? Just mass
So yeah,
provisioning?
so sometimes you have public health programs that are based on mass provision of antibiotics to, in a way, you try to prevent certain infections. People may not even be ill, but they receive antibiotics. So this is also something that can be considered as one of the potential drivers of antimicrobial resistance. And here we see it's antimicrobial use, like Shira has mentioned, not misuse or overuse, but just use in a preventive sense. But then you can end up with antimicrobial resistance problem as well. So these are some important points that need to be considered when we talk about using antibiotics. But if we look at the drivers of AMR, it's not just antibiotics. For example, sometimes we lack quick, accurate tests to diagnose infections, which can then have a propensity to further foster the spread of AMR and resistant microorganisms. We don't have enough effective vaccines, or even the ones we do have, there is a poor uptake of those. And of course, human travel. Today we live in a globalized world, and people travel from one area of the globe to another. They can spread these resistant microorganisms. So they're spread in the community, not only in the healthcare system. These are all, I think, important drivers of AMR on top of the overuse or misuse or just use of antibiotics.
Shira, talk a bit about the problem of the prevalence of antibiotic-resistant infections in hospitals.
Yeah. The problem is that it is so prevalent that we see a patient with an infection and we must cover them with antibiotics, what we call empirically, before we know what the organism is. Cultures, unlike other lab tests, they take time to grow, and only once you grow them can you then test them for what genus and species of bacteria do you have, and then what antibiotics will work against it. It can take usually about 3 days to get that answer. In that time, you need to give them something. When you have seen over and over again that the bacteria that your patients have been growing are resistant, you are very much inclined to give them the broadest spectrum antibiotic possible so you don't get caught with your pants down, not treating the resistant organism. And so my role in the hospital is antimicrobial stewardship, making sure that antibiotics are being used in a prudent fashion, right dose, right selection of antibiotic, right duration, right diagnosis. And what I fight against sort of all day long is clinicians wanting to be sure that they don't get it wrong will use overly broad or very broad antibiotics so that they don't miss the organism. And yet, what we need to do, and it's difficult, is we need to try to keep antibiotics as narrow as possible using statistics, sometimes missing the organism, but trying to mostly capture the organism that the patient is likely to have. So broad spectrum versus narrow spectrum. Broad spectrum covers a lot of organisms, a lot of genus and species combinations, and even if they happen to harbor resistance mechanisms. Narrow spectrum means it might only cover the most likely organism that patient has given their most likely site of infection. And every single time you prescribe an antibiotic, it's a balancing act.
So I remember once going into the doctor. I had a horrible throat, and I thought I must have a strep throat. I mean, I stuck my tongue out in the mirror and looked awfully red. And I went into my doctor. I said, Jack, I got a strep throat. And he says, well, what makes you think that? And I said, oh, I feel awful. So he in the office, he did a little swizzle with a stick and stuck it in a tube. And he said, go sit out in the office for 10 minutes. And 10 minutes later, he calls me back and he says, it's a viral infection. Go home. I'm not going to give you any antibiotics.
That's a good doctor. He knew that it was likely to be viral and he didn't give you any antibiotic. On the other side of the spectrum are doctors that would say, okay, well, you could have a virus, you could have strep, or you could have something else. So instead of nothing or penicillin for strep, I'm going to give you a broad spectrum antibiotics like a Z-Pak that covers more things because I don't want to get it wrong and I don't want you to get sicker and I don't want you to come back. And what they, the physicians these days, prescribers often say, I'm going to do this just to be safe. And what they fail to realize is that Giving overly broad antibiotics or giving antibiotics when they're not indicated isn't the safest option. And this is really a behavioral science thing. It's a psychology thing. What makes us prescribers say the safe option is to give you too much treatment? The safe option is to give you something that I know is going to promote resistance. Why isn't the safe option to give you nothing and see how you do for a couple of days and have you call if you're not well?
Well, let's come back to some of these very practical grassroots, down home ways to approach antibiotic resistance. But Tomislav, can you tell me a bit about briefly, it was a large study published in the Lancet, the global burden of antimicrobial resistance, studies done around the world. Tell me about this study and what the results were. I gather the highest burden is of antimicrobial resistant infections are in West Sub-Saharan Africa. But tell me about this study briefly and what it found.
Yeah, thank you, David, for highlighting this study and this paper, because it is today the most comprehensive approach to assess the global burden of AMR. Before that, we had a frequently cited review on antimicrobial resistance, also known as the O'Neill paper from the United Kingdom, that estimated that by 2050 we will have the loss of 10 million lives per year due to AMR. But the paper was heavily criticized. It was not a primary analysis. It was not peer review, and many wondered whether the threat here was overestimated. But then the Institute for Health Metrics and Evaluation, since they're invested in the global burden of disease study, understood that the full picture of the global burden of disease cannot be painted without the insights into the AMR problem. And this is why this huge collaboration with the University of Oxford and many other partners started. And when the paper was published, and even before that, when we had the results, everyone were astonished that the estimations reveal that almost 5 million people who died in 2019 had some drug-resistant bacteria, and that AMR directly caused approximately 1.3 million of those deaths, making it a leading cause on a global scale. And the magnitude of the problem was shown to be at least as large as some other major diseases, such as HIV and malaria, and potentially much larger, which means the figure that I mentioned from O'Neill, we are much closer to that figure than we previously thought, and that over 1 million people per year are already dying due to infections such as lower respiratory, bloodstream, and intra-abdominal infectious syndromes that are further complicated with the AMR.
And for some of the microbiology wonks out there, what are the names of some of these bacteria? Can you just briefly rattle off some of the names of these bacteria?
Yeah, of course. So first, I would like to emphasize that this study estimated both pathogens and specific pathogen-drug combinations. So when we look at the separate pathogens, 6 of them were each responsible for more than 250,000 deaths associated with AMR. And these were the Escherichia or E. coli, Staphylococcus aureus, Klebsiella pneumonia, Streptococcus pneumoniae or strep pneumo, Acinetobacter baumannii, and Pseudomonas aeruginosa. And on the 7th place, it's also good to emphasize it was a Mycobacterium tuberculosis, the causative agent of tuberculosis. And within these pathogens, of course, some pathogen drug combinations are very pervasive. Like you said, MRSA, and this is methicillin-resistant Staph aureus. Or, for example,
so Staphylococcus aureus, it's resistant specifically to methicillin.
Yeah, and by that we mean also then that this resistance is these strains are resistant to many beta-lactam antibiotics. So some of these beta-lactam antibiotics were the first one to be introduced into the clinical practice historically, and now we see that we are losing the battle here, and many pathogens are resistant to them now. And also
these are the penicillins.
Yes. And this study showed, for example, even E. coli is very resistant to aminopenicillins, the high burden of that, which is also a similar, the same group. So we see we lose a lot of antibiotics here, and this study has shown that. And on top of that, what's important to emphasize is that out of these 7 deadliest bacteria that I have mentioned, vaccines are currently available for only 2 of them, for Streptococcus pneumoniae and for Mycobacterium tuberculosis. Of course, there are some vaccines in development, and there is a problem because some of these bacteria can be a normal flora, like part of our microbiomes. So it's sometimes cumbersome to develop vaccines against them. But yeah, we have to be cognizant. These are huge numbers, and now we see the threat is real. The pandemic is not silent anymore. It has a true voice. And
where's the burden the greatest? Where in the world are people most dying the most from these drug-resistant infections?
yeah, so it was really sobering to see that the sub-Saharan Africa and South Asia face the highest burden of AMR. And also, one of the most worrying signs was that AMR disproportionately affects young children. So in 2019, 1 in 5 deaths attributable to AMR occurred in children under the age of 5, especially in these regions. So this is very problematic. We also found very high AMR burden in Eastern Europe and Southern Latin America regions. And maybe on the first glance, it seems counterintuitive. We would expect that when you have a high antibiotic consumption in high resource settings, the burden would be disproportionately higher in these settings. But as mentioned, the rates are highest in sub-Saharan Africa and South Asia. So we here have to observe the bacterial AMR burden as a function of both prevalence of resistance and the underlying frequency of these critical infectious syndromes in these regions. In other words, there are very, very high rates of infection in these regions. So of course, high rates of AMR will also be a problem in these regions. And of course, when you
So in sub-Saharan Africa and South Asia, you get lots of people getting infected with all sorts of things, respiratory infections, gut infections. There's a larger total number of infections. And so although antibiotic use is less, you're going to get a higher absolute number of drug-resistant infections.
Yeah, that's exactly it. And although also the fraction of the resistant infection is also high in these regions, so and they further complicate the clinical course of the infected individuals. So this is a problem that will have to be addressed definitely in the future.
Ishira Doron, you've written with a pair of colleagues, quote, the COVID-19 pandemic has revealed the deadly impacts of structural racism and systemic health inequalities on racial and ethnic minorities in the USA. Can you super quickly elaborate on this vis-a-vis COVID, but then make the leap to drug-resistant bacterial infections? That these infections, I mean, COVID, neither COVID nor drug-resistant bacterial infections are affecting populations in the United States equally. That Black and Latino and Latinx populations are seem to be the most susceptible. Talk about this if you can.
Yeah, I mean, it didn't take long for us to figure out that something was happening here with COVID and inequality. I don't think we really expected it. Right from the beginning, we were seeing that Black people were dying at over 2 times the rate of white people, and that Latinx people, they couldn't stay home for the most part. They were working outside the home in essential jobs that can't be done from their laptop at a greater rate than white people, and they lived in certain communities where cases of virus were just exploding. And so we started to think, we work on AMR, and that is the next pandemic, and that is also killing people at a high rate. And yet, although there are geographic differences in AMR that we just spoke about, and in morbidity and mortality due to AMR, what about when you're talking about our country, our wealthy country for the most part, one sort of geographic area that has minoritized individuals within it? Could it be that, like COVID-19, we have disparities in terms of rates of AMR and in terms of the impact of AMR? And so we wrote this paper really as a call to action. And what we did is we came up with essentially 8 reasons why we thought it was entirely possible for AMR to have issues of inequity related to racial and ethnic minoritization of certain groups. And I can list what those are real quickly.
Please do.
Living in crowded or multi-generational homes. That was a huge driver of COVID-19 morbidity and mortality. And stands to reason could also be a driver of spread of AMR. Differences in non-prescribed antibiotic use, and this is particularly in our minority populations that where they or their parents or their grandparents come from other countries where it is common to take antibiotics without a prescription. Differences in socioeconomic status, we certainly know just in general, drive a lot of differences in health outcomes. Foreign travel to regions with high burden of resistant infections, again, amongst our minority populations that are minorities because they come from other parts of the world. Differences in health literacy, which are really enormous between different races and ethnicities, and how that affects their understanding of antibiotic resistance and therefore how they access and utilize the healthcare system. Barriers to accessing medical care, and differences in prescribed antibiotic use, namely how the prescriber views the patient and how they view different patients differently, and how that might impact how likely they are to give that person antibiotics, since we spoke about the fact that some people who don't need them actually get prescribed them. And then lastly, employment in food animal production or meat processing, which we actually know to be sort of hotbeds of antibiotic resistance because of what we talked about, antibiotics being used very extensively in food animal production. We're really just scratching the surface here in terms of what may be the truth, and it really has barely been studied to date.
And this is a point that you raise in your letter, that you're trailblazing in this area. There's been very, very little published in the medical literature about the disproportionate prevalence of antimicrobial resistant infections and COVID infection, I guess, in marginalized populations. Why is this? Why is there so little published in the literature about this?
I think that we weren't thinking about it. It was, like I said, it was surprising to me that COVID outcomes were so different. And then it was really just a light bulb that went off to try to apply those same concepts to AMR. And I think that we are going to need to apply that same lens to many public health problems. Some of the things that I mentioned that could be drivers of differences, inequities in antimicrobial resistance. We know that there are differences. So, employment in food animal production, we know that those people are more likely to be from the Latinx community. Differences in how prescribers view patients and treat patients by race. There's plenty of literature saying that there are differences, but no one's taken it to that next level to see if that affects AMR. So there's just a lot of opportunity here for future study.
So this kind of brings me to my last question: is what do we need to do to tackle this global infectious crisis? Is a huge question. I guess we've got to do everything. We've got to pull all the levers, but perhaps a good place to start is to try to reduce social inequities. And by reducing social inequities in the United States, in North America, and globally, that we can actually reduce the burden of this crisis, and not just vis-a-vis, bacterial resistant drug infections, but just boosting health, just generally speaking, by eliminating social and economic disparities. Thoughts on that, Tomislav?
Yeah, definitely. I completely reflect everything that Shira has said. I'm also aware that even CDC now has a nice programs in place to tackle health equity issues across antibiotic-resistant threats. For example, they have the Project First Line to address training gaps. They have antibiotic resistance laboratory network to analyze patient demographic data alongside laboratory test results in order to give a full picture, and also some other programs. So this is important. Even in the global burden of antimicrobial resistance study, What is the problem are these patient-level data that link antimicrobial resistance to patient outcomes. And I will be the first proponent to try to implement also these important socio-demographic factors also to be analyzed. And in our new cross-country analysis studies that we are pursuing now, we also are looking at AMR in the context of, for example, socio-developmental index and some other important factors to see how the countries differ here, which in a way will be a way forward on how to approach this issue from this perspective as well.
Shira, final thought on this?
Yeah, I 100% agree. We need to tackle this problem at its root. We need to work on the social inequity problem. I think that here in the United States, we have an opportunity to try to improve access to healthcare, regular preventative healthcare. Everyone should have a primary care doctor, and they don't, and that is not distributed equitably. Those primary care providers should be properly educated, both in issues of diversity, equity, and inclusion, and in issues of AMR and how to prevent AMR and how to use antibiotics prudently. There are many things that we can do here that would have an impact, and we just need to start tackling it.
Because it's a frightening prospect, like going back to where I began. We kind of take for granted one of the benchmarks of global development in the 21st century is great accomplishment of having reduced the global burden of infectious disease, and people don't die of infectious diseases anymore in the so-called developed world. They die of lifestyle diseases, heart attacks and atherosclerosis and all those kinds of cancers, but they don't die of preventable infectious diseases anymore. Of course, we know they do, approaching, now surpassing 1 million deaths from COVID. But we've kind of taken this for granted that we beat infectious disease. And with the advent of, drug-resistant infections, we're kind of this new era is looming where we're going back to a period where a simple infection turns out to be fatal.
We call that the post-antibiotic era. So, the extreme is a simple scratch could kill you. But imagine that we were in an era where performing surgery was too dangerous because we can't prevent a post-operative infection, or doing a transplant or giving somebody chemotherapy is too dangerous because those things suppress the immune system and allow infection, and we don't have antibiotics to treat those infections. We would really have reversed so many decades of medical progress if we get to a post-antibiotic era.
Tomislav, final thought?
Yeah, I agree completely. And I think from my perspective, this is important to emphasize that we need to continue with this stringent surveillance and monitoring the global burden of disease, at the same time addressing this intertwined nature of AMR. This means encompassing AMR in human health, animal health, in the environment within the framework of One Health. And this also means multi-sectoral partnerships between the research community and other experts, many experts like physicians, pharmacists, public and global health experts, veterinary specialists. And global actions should consolidate access to effective antimicrobials with infection prevention strategies, with a responsible use of existing antimicrobials, and bridge all the domains that I have mentioned in this One Health approach to this important issue.
Shira Daron is an associate professor of medicine at Tufts University in Boston and the director of the Antimicrobial Stewardship Program at Tufts Medical Center. She co-authored a letter to the journal Nature Medicine entitled Antibiotic Resistance, a call to action to prevent the next epidemic of inequality. Tomislav Mestrovic is an associate professor at University North in Croatia and a scholar at the Institute of Health Metrics and Evaluation at the University of Washington. For more information about drug-resistant infections, go to greenplanetmonitor.net. It's a living web beneath our feet, a tangled network of microscopic tubes weaving through the soil, pulsating with nutrients, tying the roots of trees and other plants into vast networks. Mycorrhizal fungi, they're called. Most plants rely on them entirely, but mycorrhizal fungi do more than just feed plants. They cycle carbon from atmosphere to soil, regulating Earth's climate, and they face a host of threats. Soil scientists have come together to protect fungal networks and are calling on citizens to help out.
Well, there are clearly some roots here that I'm digging out. I'm just trying to find some that still have the like the finest root tips attached. I'm Cas Verbeek. I'm a student at the University of Amsterdam, and I'm doing my master's thesis researching mycorrhizal networks in urban environments. So we've picked a couple of street locations and one in an urban park and an urban forest. And sort of we're investigating how that different degrees of urbanization impacts the networks and the composition of those networks of mycorrhizal fungi. We're now on the Keizersgracht in Amsterdam, which is one of the canals in the center. And yeah, we're investigating how the networks of the mycorrhizal fungi in these trees look compared to the ones in more natural locations.
Yeah, so this is the stuff we are looking for. These tiny roots. It's very, very fine. And so this is where the mycorrhizal fungi connect to the trees. We cannot see it with our own eyes. If we would have a microscope, we might be able to see some like fungal colonization. But yeah, we will get the DNA out of these roots and then see which fungi are growing inside of the roots.
Can you introduce yourself?
Yes, of course. So my name is Vincent Merks. I am a researcher from Naturalis, which is the Natural History Museum of the Netherlands. But I'm also affiliated to the University of Amsterdam. Here we are doing a project in collaboration with the city of Amsterdam. And we are trying to understand how these in this case, these ulmus trees, how they are like linked with these mycorrhizal networks to each other and potentially to other plants. Well, here there is not many other plants, but we've sampled before in the Vondelpark and more in a forest setting. And we try to understand how these plants are linked below ground by these mycorrhizal fungi.
What are mycorrhizal fungi?
So this is a particular group of fungi that are mutualistic partners of trees. So they collaborate with trees and other plants, and they help the plants to take up nutrients from the soil. So plants have roots, they can take up nutrients themselves, but nutrients are kind of scarce in many soils, so they need some help, and they get help by these fungi. Fungi can make very large networks in the soil. They are very good in extracting nutrients, phosphor, nitrogen, and they transfer these nutrients to their host trees, in this case, host plants. They don't do that for free. In return, they get carbon from the plants. Sugars, yes. So the plant performs photosynthesis, so it uses light to grow. It produces these sugars, and some of these sugars transfer some of these sugars to these fungi, and in return they get these nutrients from the soil. So it's really like a network. It's like of trade.
In the Netherlands, about 23% of the species are fungi. But of course, the species on their own are nothing. It's really about networks, and nature is the ultimate networker, I think. And somebody who knows a lot about that is our next speaker, Toby Kiers, who is professor of evolutionary biology at the Free University of Amsterdam. And Toby has worked a lot on fungal networks, has also started, how crazy can you be, the Society for the Protection of Underground Networks, very clever, and you'll tell all about these kind of things. So I'm very pleased that you're here, and please take the floor.
Great, thank you. So I co-founded SPUN, which stands for the Society for the Protection of Underground Networks. And it's a nonprofit to map and protect the Earth's underground fungal networks. So when we talk about underground fungal networks, we're talking about mycorrhizal fungi. And mycorrhizal fungi are a class of organisms that form a symbiosis with plant roots. And they forage in the soil for phosphorus and nitrogen, and then they exchange these resources for carbon from the host plant. So basically plants are covered with these fungal networks, and they're feeding them carbon that they fix from the atmosphere, and they feed it to arbuscular mycorrhizae in the form of sugars and fats. And so the fungi can't live without this carbon, and so they rely completely on their carbon source from these roots, and in exchange they're providing phosphorus and nitrogen that allow the plants to grow big and healthy. Now there's billions of these hyphae under an acre of forest, and there's tens of billions of these hyphae under an acre of grassland. So anywhere between 30 and 50% of the living biomass of these soils is these arbuscular mycorrhizal fungi. Now, I think a lot of people don't understand just how much this leads to intense carbon stocks underground. So understanding the underground is really paramount to our future. Anywhere from 30 to 50% of the living biomass of soils can be made up of these fungal networks. So just to give you an idea of how massive that is, if you're to take, let's say, a hectare of grassland and lay out all of the fungal network in that hectare as one long line, it would be equivalent to about 12 million lengths of the Amazon River in 1 hectare of grassland. So again, just an enormous underground nutrient system below our feet that's just pulsating with carbon and nutrients and consistently flowing. You have to remember these are microscopic rivers of nutrients, and they're flowing really fast. So inside a fungal network, it would feel like you are moving about 50 kilometers an hour, 30 to 50 kilometers an hour. But that's just the average speed. They can get up to speeds that are really, really quite high. So it's hard to imagine what it's like to be inside a fungal network. But basically, it feels like you're moving inside a pretty, a steady moving car, I would say. The fungi, they have to be moving carbon in one direction and phosphorus in the other. But exactly how they do that is still completely unknown to scientists. So somehow they are separating these resource streams without any sort of barrier, no physical barrier that breaks it down. So imagine, it's sort of like you're having traffic, but like not having any lanes where you're going one way or the other, but just self-organizing these waves of resources. And so what we're doing is now tracking individual particles that are inside these fungal networks and trying to understand how they're using these flows to move the resources in a way that not only allow them to extract the resources and trade it with the root, but get a fair price in the sense that they are essentially calculating across the entire network where to move the resources.
And they're vast. Individual mycorrhizal fungi form vast networks and extend over huge areas, and they are individual, single individual organisms.
Yes, so they are individual organisms which potentially can cover, maybe even square kilometers in forests. We actually do not know. But so the idea is that, yeah, they make these networks, they grow these individual fungi, but it's always like it's a community, it's with many different species, and they simultaneously link different plants. So they create really like physical links between plants below ground. And they really, they trade these sugars for these nutrients in the soil. And there is some very interesting research by Toby Kears that really, the fungi can really choose which plant they give more nutrients, and that of course they give more nutrients to the plant from which they get more sugars. And the other way around is also true, that the tree is able to give more sugars to the fungi that give them more nutrients. So it's really like a trade interaction.
My name is Toby Kears. I'm a professor of evolutionary biology at the Free University in Amsterdam. The fungi are actually very good at taking resources from where they're very readily available and then moving them across the network to places where they're less available and the plant is willing to pay more for them. So where resources are low, they are getting more carbon in return from the plant. And you can imagine, I mean, these fungi have been under natural selection for hundreds of millions of years. It's not surprising that they would have evolved strategies that allow them to optimize their trading patterns.
It's really a bit about a poker game, right? So this is what we're trying to understand, is this trade for carbon to phosphorus, and you've got the plant root on one side and the fungal network on the other, and they've been doing this for 450 million, I get shivers thinking, 450 million years. And so this is me in the corner, just trying to watch. And I would argue that a lot of what's been happening is in the dark, right? In these systems, it's totally underground. So it's very, very hard to understand and track these trade strategies.
So I'm opening up a video right now of some of the dynamics that we're seeing inside these networks. What surprised people the most is that this is in real time, right? These are videos that are not sped up. This is actually what's happening below our feet. And so it's a very, very fast, dynamic movement. It looks like a living river, but how can a river have so many different dynamics at once, where you can have the river banks flowing in two different directions simultaneously? Nutrient rivers that are just pulsating with carbon and phosphorus and nitrogen, and they're connecting plant roots. These are ecosystem engineers. I think most people would agree now that if we lost the fungi, the soil would not stick together. It would just start washing away. Fungi are the seam that hold ecosystems together. And yet we don't know who is where. And so we've started this project as the first attempt to get a global picture of where these fungi are. In the next 18 months, we're trying to gather about 10,000 samples. And so it's a massive effort to put these fungi on the map, the way that we have maps of global vegetation or ocean currents, right? These are all systems that we have started to map. Even the stars we have better maps of than global fungi underground. And this is just key. It's just shocking to me that we haven't developed these maps, considering how important they are for our climate, how important they are for plants and therefore for animals, all surviving above ground, and yet to have no idea of how these networks connect ecosystems across the earth is shocking to me.
So what is known is that some of these fungi are extremely sensitive to pollution, for example, nitrogen deposition, because they help the trees to take up nitrogen. But if there is enough nitrogen in the soil, the tree is going to say, oh, I can just get it myself. I don't need these mycorrhizal fungi. So what you see is if there is a lot of nitrogen deposition, which is a huge problem in Europe and around the world,
This is deposition from the atmosphere.
Yes, from the atmosphere, or it comes from agricultural lands. It goes into the water, and so we add a lot of nitrogen basically to our environments, and what you see in places with high nitrogen, the mycorrhizal community is much less diverse. So there was just some generalistic species, and you lose some of the probably more specialist species. So in that sense, we need to protect what is underground.
There's a really large and growing body of research that's showing just how negative deforestation can be for fungal networks. And sometimes it takes decades for these networks to recover. Fire systems, there's some fungi that are amazing that are able to come right back after a fire system. But these symbiotic mycorrhizal networks are not one of those types of fungi, and they take a very long time to come back. And again, decades can go by, and then still low levels of diversity, low levels of abundance. Agricultural systems, especially conventional agricultural systems, where there's tillage, there's pesticides, there's fungicides. Eventually there's just not much soil life left. I think the latest UN numbers say that 90% of soils will be degraded by 2050, right? 2050, I can picture 2050. That's very soon. And so that's what we have to worry about. Don't leave your soil bare, right? That's a really, really, really bad thing for fungal networks. Advocate for green roofs in cities. A lot of people don't know, but these fungal networks, they spread through spores airily. They're in the air, the spores, right? And so if they can have some kind of corridor of green through cities, this helps them survive. This helps them form healthy networks within the urban environment themselves, is having green spaces. So there's people can talk about underground ecosystems the way that they talk about Amazon forests and coral reefs, right? They can start to appreciate the organisms that live down there. And I think to be able to do that, they need to understand, they need to start being able to visualize what is under our feet.
So we found it's fun to start to map, protect, and harness these fungal networks. What we want to do is get citizens and the public and activists involved in this process. People that are interested in fungal biodiversity and are interested in going out and sampling in their own yards and helping us build a bigger, clearer global picture of these fungal networks. In the next 18 months, we hope to collect about 10,000 samples. We want to develop the first red list, right, for fungi. Which ones are we most worried about? And maybe not within the first 18 months, but a big goal of ours is to develop the first fungal conservation easement, right? So that we could have land conserved for something other than plants or animals or the things that you see above ground, but a conservation priority for something you don't see below ground. And I think this is kind of a moment, right, where we have this chance to overturn our above ground bias, right? I mean, for decades, we've been focused, centuries, everything is about above ground climate solutions, above ground conservation strategies. All the agendas are about what we see above ground, but they fail to recognize what's happening below ground.
Yeah, this initiative that Toby started, right? The Society for the Protection of Underground Networks. I mean, yeah, we really need to map these networks globally. We really need to try to understand, what these networks look like and how they function ultimately. Yeah, it's really exploration, and it's essential. Without these fungi, the trees wouldn't be able to grow. And many of our terrestrial ecosystems would simply not exist the way we know them. So the real challenge here is actually to get to the roots. So you have to break through the pavement. This guy from the city that's going to help us has brought some tools, and they're now lifting some of these stones from the pavement. And it's intriguing, like there are people walking here and cars parked, but we are really walking over a living network of tree roots and fungi. There's like so much happening below our feet, even in the city, you know, that is remarkable, I think.
Underground networks.
Yes, underground networks, yeah.
Special thanks to Bart Brown and the Naturalis Biodiversity Center in Leiden, the Netherlands, for the audio track of Toby Kears' presentation. Learn more about mycorrhizal fungi and the underground networks they form at greenplanetmonitor.net. That's it for today's GPM, coming to you from Earth Chronicle Productions, Breda in the Netherlands. Listen, read, and watch more Earth stories at greenplanetmonitor.net. I'm David Kattenberg. Over and out for now.